Hello, everyone. Today we kick off Beckman Foundation's Milestone Moment celebration of the 100-year wedding anniversary of Arnold and Mabel Beckman. It's my pleasure to introduce our guest speaker, Dr. Yevgenia Kozorovitskiy. She was named a Beckman Young Investigator in 2015 and currently is the Irving M. Klutz Professor and an Associate Professor of Neurobiology in the Weinberg College of Arts and Sciences at Northwestern University, where her lab is broadly focused on mechanisms of neuromodulation and neuroplasticity, synergizing biological discovery with optical and molecular methods development. So today's presentation is a synchrony of minds and brains. And so this is how dopamine and oxytocin collaborate for joy and love. So Yevgenia, welcome.
Thank you, Carrie, for that kind introduction. Let me share my slides. And welcome, everybody. I'm truly honored to kick us off for the scientific part of our celebration of the love and life of Dr. Arnold and Mabel Beckman. Here on the right, you see a picture of Arnold and Mabel. They are hiking in the Valley of Death in the 1920s, probably just a little bit before their marriage in 1925. The best of friends, soon to be the closest of collaborators, and an inspiration for us all with their love so deep that it outlasts life itself. Social relationships lie at the core of our beings. And not just you and me, but everywhere in the animal kingdom, we can see social bonds of romance, parenting, sibling love, and friendship. Even DALL-E 3, the OpenAI text-to-image model, does a pretty decent job of capturing many essential social behaviors across the animal kingdom. And all these complex social interactions, just like our most fundamental needs and drives to sleep, to eat, to move, are profoundly governed by neuromodulatory chemical signals inside our brains. And this is true for many important learned behaviors that we see across the animal kingdom, as well as those that are innate, that do not need to be learned and are profound, important, powerful drives. So in the lab, we're able to capture some elements of those behaviors and their underlying biological mechanisms using animal models. This work is important for understanding the brain and how it works, but it also matters enormously for being able to help people who may have problems with social relationships. For example, mothers who may be suffering from postpartum depression, or folks that are dealing with complicated grief. I'm really happy to be able to share with you today a little bit about the studies that emerged from my 2015 Young Investigator Award. And this is also a little anniversary for me today. And this work is on the synergies between the neuromodulators, oxytocin and dopamine, two really important chemicals in our brains. Oxytocin is a small peptide that's synthesized in a part of our hypothalamus in the paraventricular nucleus and supraoptic nucleus. You can see marked here in purple. And it activates a G protein coupled receptor in our neurons and in other cells throughout the body. Really important peripherally for such functions like conception, giving birth, the induction of lactation, and in the brain, indeed, for all of these social, positively valenced social interaction behaviors. And so that's the brief introduction to oxytocin. And now dopamine is another really critical chemical in our brain. Here you can see its tiny structure. It's a monoamine, and it's synthesized also deep in the brain in a few different areas that you can see in this example, coronal section through a mouse brain, in a part of the brain called the ventral tegmental area, and substantia nigra pars compacta, fancy Latin names. And different groups of neurons that synthesize dopamine are really important broadly for controlling reward-based behaviors, but also in substantia nigra for regulating locomotion, complex motor tasks, and goal-directed actions. And dopamine activates two different classes of membrane receptors that are separated into D1 and D2 type dopamine receptors. So it is intriguing to consider just how those two systems may potentially interact. And indeed, it has been realized for a while, in part using this really cute little animal called the prairie vole that shows really strong social bonding patterns, that dopamine and oxytocin systems can talk to each other indirectly. And later work has found some evidence for that in mouse models as well. And I've seen even glimpses of that in my graduate research, looking at close parenting behaviors of a biparental small primate called marmoset. So this early work demonstrated that oxytocin system and dopamine system can converge in a part of the brain called nucleus accumbens that's important for reward. And work that emerges from my Beckman Young Investigator Award also showed that they actually can interact directly. Oxytocin can talk to dopamine neurons right in the place where they reside. And then I'll share with you also a little bit of more recent work where we're exploring the interactions of those two systems in medial prefrontal cortex, which is the frontal part of the brain that's really important for higher order cognition and also, no surprise, for important social behaviors. So this work began really right around the time, just a little bit before my Young Investigator Award, with a very talented first postdoc in our lab, Xiao Lei, who recently got tenure at Fudan University. And he started with just a very careful anatomical mapping of the oxytocin system and quickly discovered that, in fact, oxytocin neurons sent projections that you can see here in green to the sites where dopamine neurons reside. And he did this backwards and forwards and in all sorts of different ways to convincingly demonstrate that the anatomical substrate for the interaction is there. And then he proceeded to record from dopamine neurons to ask, are they in fact modulated by oxytocin? And dopamine neurons like to be tonically active. And I'm showing you a recording from the ventral tegmental area with a histogram of activity and some insets for times 1, 2, and 3. And you can see that the dopamine neuron is a little bit spontaneously active. Each of these vertical dashes is a spike, the primary language by which neurons talk. And you can see that after the application of oxytocin, even a brief one, the activity of this neuron goes up. And then later, when oxytocin is gone, it goes back down. Interestingly, Lei observed the opposite effects on dopamine neurons in substantia nigra, where the activity of a neuron declined when you applied oxytocin, and that was later recovered after washout. And this is really important because the dopamine neurons that are on the left are very important for encoding reward, while those on the right, their activity facilitates movement and exploration. Thus, the pro-social peptide, the love hormone oxytocin, can bias dopamine tone in the brain towards rewarding things. Next, we wanted to ask the question, well, in those axons, is there actually enough oxytocin in order to provide the neurobiological basis for this kind of effect in the living animal? And for that, we turn to optogenetics. So here you can see the heart-shaped hypothalamic nucleus, and in red are all the oxytocin neurons. And for this experiment, they're expressing a very special opsin that allows controlling cells with light. So this seems a little bit science fiction-y, but similar kind of technology is already in use today in the context of retinal prosthesis to cure some forms of blindness. And so we were able to do this experiment. You can see here in the white are some dopamine neurons across the two brain regions of interest, and these delicate axons of oxytocin cells that are seen throughout the images. When we were able to recapitulate the prior observations to show sufficiency of oxytocin that's locally located in order to activate dopamine neurons in the VTA and to silence the ones that are less related to processing reward. And we're able to demonstrate the full circuit by which this occurs, where the enhancement of tonic activity of VTA dopamine neurons is a direct effect of oxytocin activating receptors on those very neurons. And indirectly, you have the silencing of the activity of substantia nigra dopamine tone. And at around the time that we published our work, another study came out that clearly also demonstrated that oxytocin release in the ventral tegmental area can enhance prosocial behaviors. In studies that I don't have time to describe to you today, we also found that oxytocin not only biases dopamine tone in the brain, but it also changes what dopamine neurons listen to. The way this works is that oxytocin receptor on dopamine neurons causes the production of endogenous cannabinoids in our brain that then travels presynaptically and can modulate release from the glutamatergic terminals that are activating those neurons. And so that's quite interesting. And we found that this mechanism has specificity for different pathways that feed into dopaminergic neurons. And some really important for life inputs actually bypass this kind of filtering. Thus, the interactions between dopamine and oxytocin are really quite complex. And so having recognized this for dopamine neurons themselves and other neurons that reside nearby, as well as the inputs to them, we were wondering, well, is this really it? Could we be missing potentially other important interactions? Because it seems so compelling from evolutionary standpoint that the systems that support affiliative social behavior, that support things that we think of as love, really must involve some kind of a tapping into the conserved neural circuits that broadly regulate reward. And so to start off this work, Sarah Freda, a graduate student in our lab, decided to take a very careful brain-wide look at all of the oxytocin system, all of its inputs and all of its outputs. This is the kind of work that we cannot do today with this level of precision in the human brain, but is possible in other species. So here she expressed the fluorescent protein in our little heart-shaped nucleus, and then looked brain-wide and carefully, quantitatively mapped all of the places where oxytocin neurons from this nucleus project. And she did the same thing for the inputs that these neurons listen to. And this is example of some of the data for the brain-wide mapping. And in the part of the brain called the cortex, so the superficial parts of our brain that are so strongly involved in humans, we found that the maximal amount of oxytocin projections was in the part of the brain called prefrontal cortex. That's really important for regulating social behavior and generally complex cognition. So here you can see some example axons in the coronal brain section. This clearly pointed to prefrontal cortex as another likely place where dopamine and oxytocin may collaborate, because you can see here locations of the dopaminergic axons and the oxytocinergic axons that are localized to the same part of the cortex. And diving in further, we even find that if you look at individual neurons deep in these cortical layers, many of them express both oxytocin receptor and dopamine receptor, here seen as little puncta that represent mRNA transcripts for those receptors. So you have both at the level of brain region and at the level of single neuron, the possibility for convergence of oxytocin and dopamine, thus providing a really concrete, even single cell level substrate for the encoding of social reward. Sarah is now carrying out a lot of electrophysiological studies that show that, in fact, oxytocin does modulate neurons that also listen to dopamine. And she can do those studies both at the single cell level to be able to have deep mechanistic insight into just how this collaboration occurs, but also at the level of circuit using in vivo extracellular electrophysiology. So I've showed you today a little bit of data about how oxytocin can modulate excitability of dopamine neurons, how it shifts the bias in the activity of different kind of dopamine neurons, and can even filter the sorts of inputs that they care about. And now we're doing a lot of interesting work in prefrontal cortex on the excitability of dopamine sensing neurons by oxytocin and characterizing the neural circuit motif within the cortex that can further support the mechanisms that regulate social behavior. Emergent out of my young investigator work is now a large collaborative grant that's funded by the NIH Brain Initiative that looks at interbrain synchrony in two different rodents, mice and prairie voles, that have diverged many millions of years ago, and they differ in the ways that they express social behavior and affiliative behavior, and in the way that they bond. And broadly, in this project, which is a close collaboration with the lab of Zoe Donaldson at CU Boulder, we hypothesize that oxytocin and dopamine interactions in prefrontal cortex are a key driver of biobehavioral synchrony. To tell you just a little bit more about this, I'd like to explain what interbrain synchrony means, because it's kind of, frankly, a strange concept. Interbrain synchrony, by which I really do mean synchrony across two different brains, can arise from ongoing social interactions between animals. It was first actually discovered in humans doing what is called hyperscanning paradigms, where you can measure neural activity from more than one person at once. It's observed across broad range of frequency bands, depending on the kinds of methods that have been used in order to look at this. Now, the notion of interbrain synchrony is fairly easy to grasp if you think of doing the same dance, if you think of shared language, listening to this talk, for example, I would expect a little bit of neural synchrony across all of our brains at once. But it's a bit more surprising and more controversial if you think about neural synchrony, where it's been shown in the context of open-ended, free-flowing social interactions and games, for example, that involve cooperations between individuals. And note that neural synchrony across brains can be especially strong in partners, in people who love each other, and during positive social interactions. So at the time we were, here also is just an illustration of synchrony for mice that can interact freely or are separated by a clear barrier or cannot see each other. And here in two different colors are traces from neural activity in prefrontal cortex of animals with these conditions. And you can see that correlations reported by this R value are different. So the traces in the bottom here are actually mathematically more similar to each other. So this is the synchrony during sociality. And at the time we were starting to get really interested in this question and thinking about the potential of dopamine oxytocin interactions and driving it, this question of synchrony has never been causally examined. It's just been reported across multiple species, fascinating correlative line of work. And we decided to see whether it may be possible to actually ask the question whether it is a causal contributor to positive social interactions. And as you can imagine, asking these questions with the kind of the level of precision that we can do in neurobiological studies can sometimes run into problems of tethers, right? You can see here's a picture. This is from Zoe's lab of two very friendly prairie voles that have completely knotted up the tethers through which the brain recording was being carried out. To help solve this problem for our studies, and also in parallel to facilitate the development of optoelectronic tools that may one day find usages in the clinic, my lab works closely with the lab of John Rogers, who is a materials scientist here at Northwestern. I should note that the first time I met John was actually at the Beckman Symposium around 2016 or 2017, I forget. And this accidental meeting sparked our future scientific collaboration. So here you can see one example of this tiny optoelectronic devices, which we create that are powered by radio frequency fields that can help synchronize or desynchronize prefrontal cortex using optogenetics, just as one may do with, for example, transcranial magnetic stimulation in the human brain. Using this kind of approach, we actually carried out the experiment. And you can see an example video here where during synchronized stimulation, these two mice very closely interact socially and spend a lot of time together. Whereas during a period of desynchronization, we found that they don't like to interact nearly as much. So sociality is bidirectionally regulated by synchrony. And you can see here is some summary data with various controls that I won't be able to fully describe today. Okay, so this provided the first kind of causal basis for thinking that neural synchrony is an actual contributor at the causal level for sociality. And now Isabel Reith, a graduate student in our lab, is using all kinds of different tools and AI-assisted careful behavioral mapping in order to study this further and understand what are the key factors that are driving the emergence of neural synchrony. And I want to show you here that we're able to zoom into it quite closely and see, analyze the periods and the behaviors when we can see high levels of synchrony. So here, the red line represents synchrony going up or down, and blue represents, in this case, proximity between animals. And you can see that as they get close together, synchrony is rising. And when they turn apart from each other and move away, it also falls. And so we're able to really, hopefully, with this kind of approaches, gain a nuanced understanding of just how the synchrony emerges and what it does and how it helps support affiliative behaviors. As part of this work, also in collaboration with Dan Dombeck here at Northwestern, we have complex hybrid types of paradigms where we're able, while animals are interacting freely, get really close-up looks at neural activity using genetically encoded calcium sensors, so we can truly characterize the ensembles and the contribution of dopamine and oxytocin to the actual encoding of social behaviors. So to summarize, I've shared with you a little bit of our work today on how oxytocin and dopamine systems talk to each other in lots of places and lots of different ways, with medial prefrontal cortex, which is really important for cognition and sociality, emerging as one key site of direct convergence. I've also shown you some exciting preliminary data that interbrain synchrony may causally and bidirectionally modulate sociality. And we expect that oxytocin and dopamine interactions there will prove to be very important in shaping this neural and behavioral synchrony. And this work is quite important in understanding neurobiology behind joyful social pleasure, behind trust and bonding, and also behind love, which we're here to honor today. I'd like to thank collaborators and my team, which here you can see the current members of the lab saying hi to you from the state of Illinois, where Dr. Beckman was born and grew up. And I want to also thank the Arnold and Mabel Beckman Foundation for hosting today's event and for always supporting science that helps humanity humanity and for channeling the legacy of Dr. Arnold and Mabel Beckman since the time before I was even born, both in the times of national challenges to scientific progress and in the decades of America's strong global leadership in innovation and discovery. And with that, I thank you for listening too. I'll stop sharing the slides and I'll be happy to take any questions.
Yevgenia, thank you so much for that very interesting and informative presentation. Thank you so much. We are open for questions with Yevgenia. If you have a question, you can put it in the Q&A function that you have at the bottom of your Zoom screen, or if you like, you're free to turn on your video and unmute yourself if you'd like to speak directly with Yevgenia at this time. So we can do that now. We can take a couple of those we have time to do right now. Yevgenia, I will say that when I saw the video of the mice with the optoelectronic tools, when you were talking about them in synchrony and then desynchronized, my mind jumped to couples counseling, just because I don't know what the sort of the long-term applied use of this would be, but to imagine the applications in that kind of a setting is kind of an interesting thing, you know, to try and, you know, synchronize maybe the thoughts, or to encourage the bonding between people who are looking to do that but aren't in sync. I don't know.
That's exactly the right direction to think about in the context of couples therapy, but also in the context of when, you know, people, for example, have problems bonding with their children. You know, there are ways to modulate neural synchrony, you know, both kind of by stimulating the brain directly. There are also, you know, shared exercise activities and things that therapists can do that, you know, I expect certainly relate to increasing, you know, indirectly, right? We have many means for doing that neural synchrony across brains. And the thought is that this kind of, you know, positive recurrent feedback between neural synchrony and positive social interactions can really help individuals that are suffering difficulties with social behaviors.
Fascinating. So we do have a question here. What sparked your interest in neural synchrony, and how did you come to see it as a key piece in understanding brain function or behavior?
Well, so, you know, honestly, it was kind of, it was almost a little bit accidental in that we really were thinking a little bit historically, as I showed in my talk today, that surely oxytocin and dopamine systems must talk together a whole lot. And so we were looking in the places where dopamine neurons reside, and then pretty quickly came to realize that prefrontal cortex may be potentially a really important site, especially in the deep layers for oxytocin and dopamine and interactions. And it's actually those deep layer neurons that are thought to be important for setting kind of oscillations in our brain. And there they were, you know, expressing dopamine and oxytocin receptors. Mm-hmm. At the same time, at that time, there was a bit of an uptick in the amount of correlative bits of evidence that support this idea that interbrain synchrony isn't completely crazy and that it can occur in these open-ended social interactions. And we realized that all of this is potentially kind of positioned for working together in this part of the brain that's important for kind of higher level functions. But we really wanted to have a causal essay for this as well. And so that's what started, like that first collaborative paper between John Rogers and our labs. And now that work continues, you know, through a variety of different separate projects, some of which relate to the question of neural synchrony and some of which relate to other important questions in the space of neuromodulation and plasticity that my lab is so interested in.
Thank you. All right, do we have any other questions for Yevgenia? All right, great. Well, thank you so much, Jenny. I appreciate it. We do have, we will prepare a recording from today's presentation. It will be available on our website tomorrow. And if you haven't yet registered for our other live presentations for our Milestone Moment celebration, we do have time still for you to register. We have another coming up at 10 a.m. Pacific time today and one at 10 a.m. tomorrow. So, Yevgenia, thank you so much. We appreciate you kicking us off today with such an interesting presentation.
Thank you so much, Carrie, and I look forward to seeing the rest of events in this series.